Sarah will tell us about what does Instruct-ERIC Finland offer to Portuguese centers. Thank you very much.
Thank you. So it's a pleasure to be here, and everything that I tell you, you should be able to find on the ARIA website. So don't worry if you miss something. I'm also here until Sunday morning, so please stop me if you want to discuss something. So, the centre in Finland is a kind of distributed centre. So we have 3 universities taking part. These are part of our national infrastructure in structural biology, which is in 5 different centres. And so I will tell you little highlights of things which I think are most useful or different to what you'll find in other centres, and so why you might want, of course, to come to Finland to do your internship or your research. So, where are we on the map? Susan already indicated that, well, we know Europe's very big, and there are 3 places which are really on the edge of Europe: Finland, Israel, Portugal. So we do understand the issues of being on the edge, but we know it's the cutting edge, isn't it? Okay. Right, so in Finland we have the various people involved. So these are our 3 centres. So we are in the south. This is up close to the border with Lapland, and then this one is right next to the Russian border. So I'm in charge of the Instruct Finland object. I'm also vice chair of the Instruct-ERIC Council, so if you have issues relating to the running of Instruct in general, come and talk to me. I'm aided and abetted by Hanna Oksanen. And within the University of Helsinki, we basically have 4 different objects which we are offering. Minna Poronen runs the biocomplex purification unit. Then Perttu Permi is running the Nmr. Markku Varjosalo is running the single-cell proteomics by mass spectrometry, and then I'm running the Microbial. And we already have, through the impact widening project, we have a lot of collaborations with Portuguese scientists, and so especially with the Microbial, we're really trying to help. So up in Oulu we have Lari Lehtio, who's running crystallography setup. He particularly also has, related to that, a lot of molecular biophysics equipment, but he's been very instrumental in the development of this thing called PCISBIO, which is a crystallographic data management system. I'll highlight that a little bit. And then in the University of Eastern Finland we have Juha Rovinen, who's also been doing a lot of work with Portugal in terms of mass spectrometry, and so he's been part of this EU network on mass spec, and I really hope that will also be renewed. So I'll also spend a little bit of time more on his work as well. So up in Oulu, I'm going to use these guides here on the slide to kind of indicate to you, if you go to the catalogue in ARIA, what you're looking for. So you're looking for sample preparation, 3D structural analysis if you're looking for Lari, because they are doing molecular biophysics, crystallization, X-ray diffraction, and then this crystallographic data management. I make the presumption that you all know what is crystallography and what is X-ray diffraction. Is there anybody in the audience who doesn't do? No hands? Excellent, okay. So I'm not going to explain that. But if you are doing X-ray crystallography, this research data management for structural biology basically allows you to design your crystallographic experiments, collect the data from the images, send your crystals to synchrotron, collect the data from the synchrotron, and is integrated into the ISBYB, which is used as synchrotrons for crystallographic data collection. So this is something which is freely available, it's easy to install, it's a good educational tool, it helps communication between different project members, it works on and off campus, and it works with quite a few different automated images. So look into that, and if you're interested, it's free software. You can pick it up, and Larry will help you if you have any problems with the installations. Going then down to, I need something to put into my crystallographic experiment or into the electron microscope, then we found it's really important to have a focus on the preparation and purification of large macromolecular complexes. So we have a very long history in virus research in Helsinki, led by Dennis Bamford. And so we have a preparative ultracentrifugation farm. I don't know if that should reproduce, maybe they should do, but anyway. Which allows for large-scale sucrose gradient purification, for instance. We have monolithic chromatography, which again really specialised in, for instance, RNA molecule production and also large complexes. And then something which you don't find in the other centres in Europe is this asymmetrical flow field flow fractionation, which is lovingly called AF4 by its friends. And this is something that we've had working now for several years. We've just had a new upgrade to it, so it's now not just an analytical machine, it's actually a preparative machine. It's really good for fragile assemblies of macromolecules. And so I will give you one slide on that. So the idea with asymmetrical flow field flow fractionation is really very similar to size exclusion chromatography. The idea is you have a flow, you have channels in the machine which allow you to then use diffusion and cross-flow in order to separate molecules of different sizes and shapes. And then this is coupled with a bunch of different detectors which will allow you to estimate concentration, mass recovery, mass polydispersity, radius hydration, radius of gyration, and so basically you can say quite a lot about the complexes that you're purifying. And Minna has been working extensively, as I say, on viruses and subviral particles, but also on RNA purification. So do come along, and you can couple this sort of technology then with going to X-ray, going to NMR, going to cryo-EM, so that you have an idea of how the sample preparation is affecting the quality of the samples that you're then going to use for the next step. So we have a lot of proteomics in Finland. I'm just going to discuss two different types. So initially we'll talk about native mass spectrometry, which is what Juha Rovinen is really concentrated on at the University of Eastern Finland. And here the idea is that this is top-down proteomics. You start with the complex and you want to know what are the different molecular states. So it fits also to our biocomplex purification as well. And then we also have bottom-up proteomics. What Juha has in Kuopio is FTICR, then there's also timsTOF, and then also this new ultra-high mass range Orbitrap. And this means that you can do ultra-high resolution mass spectrometry. You can get collision cross-section determination, so allowing you to look at shapes of molecules, and you can then also resolve complexes up to around 20 megadaltons with the machine. So these have different properties, different sorts of experiments that you might want to come out of them. I summarise that really on the next slide, where you see that the applications from the high-resolution MS are intact protein characterization, post-translation modifications, genomic sequencing. With the native MS, we're looking at protein-protein, protein-ligand, protein-metal interactions, stoichiometry, KD values, and also ligand screening. And then with the high mobility, we're really looking at separation based on size and shape information. A couple of examples from Juha, because I know you have the interest here in the research site in Portugal. So this is an example of aldolase where it's possible to separate the unfolding monomer from a folding monomer from a diamond from a trimer. So you get all of these information in one experiment. Another one, which I thought was quite interesting, was this thermokinetic analysis of protein subunit exchange, where basically one peptide has been mutated, and they've looked to see what is the competition between these mutants and the wild type in order to work out entropy, Gibbs free energy, in order to characterize the reaction of this interaction. So that was another experiment. And then finally, I thought one that might also be of interest to you is when you're looking at different ligands which are binding to a particular protein, then in this case, by doing a competition experiment, but measuring it by mass spectrometry, you can get very accurate measurement of KD. So this might be interesting to those of you who are looking into screen. What Marco is doing, apart from the very sort of standard bottom-up proteomics, is working on single-cell mass spectrometry. And so this is now back in Helsinki. Why would you do single-cell proteomic analysis? Well, there's a lot of interpopulation heterogeneity in any cell population that we see. And so in order to try to work that out, to know where your signal is coming from if you make a change, it's really quite interesting to look and say, can we pull out a particular population and see on the proteome level how this varies. And so this just would show you that you'd be looking for a particular molecule and see how the concentration varies between the two populations. This is a schematic which I probably can't explain, but which I got from Marco. So basically the idea is you want to look at a really small sample. You have carrier cells, you are going to tag the small sample and the reference cells and the carrier cells, mix them all together, and after you have labelled them individually, and then you basically have barcoded peptides where you can quantitate the differences between these populations, which allows you what you call peptide fragments to really identify the proteins present in each population and to quantify them based on what was actually present. It's a really, really rare technique in Europe. I only know one or two other labs in Europe which can do this. So again, please do think about whether your projects are suitable for that. The Nmr Center at the moment, we are offering access to an 850 and to 600. This is pretty standard NMR, it's not high field really at all. But what we do have, which might be of interest, is that we have the fluorine, phosphorus, silicon and lithium probe heads. We have cryo-probe heads, and we also have a large sample changer. And so you can do, for instance, metabolic analysis from plants, for instance, to compare what's actually happening in many different samples, as well, of course, with standard structure determination. In terms of cryo-EM, I will obviously spend a little bit more time on this because I'm completely biased in this area. So we have 25 years of experience doing cryo-EM, and so we've seen many, many different problems over the years. And so what we focus on in terms of what we give to the community is really this: how do I actually get a good sample? Many of the places where you can do cryo-EM, they say, oh, once you've got a really nice structural average which goes to around 5 angstroms, then you can come to our facility and collect data. We don't say that, we say, come to us with your biological problem and we'll try to work out a way in which you can get a decent sample. And then we can check that and try to develop it. These can be long-term projects, and they can be very tricky. And then at the end of the day, somebody will come along and say, oh, it's a beautiful structure, but there's a lot of work before that. And that's where we try to really help with our expertise. And so Sarah and Jose have both been, for instance, in our Optica room in Helsinki, where they've been learning electron microscopy, and they've been looking at their samples which they've been trying to actually optimize for then using these at another center. What we have at the moment, which is perhaps more unique compared to other centers, is of course we have the biocomplex in the same building. So that means you can do all of this really fancy separation technology and then take it straight into our microscope. But also, very recently, we purchased a blot-free vitrification robot, which is really good for very, very, very small sample volumes. So very often with Electron Microscopy, you say, oh, I want to use a very small volume, I've got a very precious sample, and this will actually work with nanoliter volumes. Half a micron. Okay, so we'll have a little bit of a look at that. If we could have the movie, so I'm doing a little bit of product placement here. This is the cryosol vitrojet, which this is now the second one operational in Europe. It's the only one within the Instruct-ERIC. It has a pin system, which the pins will pick up nanoliters of the sample. So we have a grid here, which is waiting for the sample. It is drawn on by this pin, very small area, which is covered on the grid. That's why you can actually make 12 samples with only half a microliter. And what's special with this is that there's no blotting involved. You only put the minimum amount of sample on the grid that you need. You don't take anything away. Why is that important? Well, the problems with making cryo-EM grids are reproducibility. Doesn't happen very often at all in terms of one grid would look like the next. With this machine, we see really good reproducibility, which is very nice. We also have tested our favourite nightmare samples, those which we know are difficult. Normal samples work fine, but what about difficult samples? So ribosomes, everybody works with ribosomes, they're easy. No, they're not. Ribosomes, you normally have to image them on carbon, which causes preferred orientation, and it decreases the signal in the sample. So these are samples prepared on the Leica, these are samples prepared on the vitrojet. In this case, we could clearly get very nice distribution of ribosomes in a standard layer of water without any carbon present, with no preferred orientation using the vitrojet. And so in this case, we've overcome some of the problems with the sort of carbon-water interface. Another exciting samples that we love are membrane proteins. Membrane proteins, especially when they're in detergent vesicles and lipids and things, they also don't behave very well in water. And we've been looking at membrane proteins in SMOPs. They've worked really nicely, no problems whatsoever. And very often with membrane proteins, you have a very small amount. They are a pain to purify, and so this is a very nice technology for that. That is actually shown in these images. You can't see much at all, but the little dots in the background are what we're interested in. We've also looked with Francisco from Porto, who's not here today, unfortunately. We've been looking at fibrous proteins. They've also worked. We've also looked at small soluble proteins and large complexes. They've also behaved very, very well, as we would expect them to. We've experimented with different grid types and different material types of grids, and we've really had no problems with them at all. The only thing that has failed so far, and I know that this is one of our favourite difficult samples, is envelope viruses, which those are tricky to get images of. We have to do some fancy stuff called backplotting in order to get them to reproducibly behave in cryo, and those don't behave. So we haven't managed to overcome this, which is a pity because a lot of our work is on envelope viruses. And then the machine also has within it the ability to do plasma cleaning, which prepares your grids for the sample. And at the moment we are destroying very thin layers of carbon which are on the grid, and so we're optimizing them at the moment. I'm sure we will get around this. And then finally, what I've been trying to emphasize is sample volume. In the vitrojet, we can use half a microliter of sample to make 12 grids, whereas normally we use 3 microliters for one grid. And so if you're looking at very precious samples, this might be of interest. So at the moment, Vitrojet or Leica, which one is winning? They're both very, very good machines. This one's easier and faster to use. This one gives a lot of reproducibility. And finally, what do we really offer to Portuguese scientists? So I think you should really ask Andrea, Francisco, Sarah, Jose. They've all been in Finland. Francisco and Andrea both for over 3 months. Sarah and Jose for slightly shorter periods of time. So for instance, you can enjoy the delights of Tallinn, which is just across the water. And of course, you can enjoy the delights of being inside when outside is really cold. And that's why we do it right here. So here we have Sarah and Jose. Thank you for your attention.
Saving a few minutes questions. Thank you very much. So, if you have any question, please raise your hand and we'll have some people. Actually, I should say, just a moment. To any of the other previous speakers, in particular to Susan, if you want to ask her something, she will be here all the day. Okay, so feel free. And in the afternoon, we'll have more strict time, but the speakers will be around soon.
Project. So, what do you advise? It's like, do we submit? Now, it's in terms of organization for the Instruct. Do we submit like a short proposal saying that to do the preliminary results, and then based on that go to the second round, or do a big proposal already with a lot of essays?
So what I would suggest is that first you contact us and we discuss, because what we standardly do in our facility is that every time there's a new project potentially coming in, we sit down and we discuss for half an hour and say, is this the right facility for you? Are you planning sensible experiments? Have we seen anything like this before? All right, so that's the first thing to do. And then after that, obviously there are these remote access, so you could just ship us your sample, but if it's something that's very sensitive, and it's tricky because it's very sensitive, it would be important to be on site. I mean, I always offer the facilities of my lab to whoever comes by, so if we need to run something, we'll do a quick expression and so on, it's all possible. Within the Instruct process, I would say if we really think it will be quite tricky, then obviously an internship would be the best way to go. But we have also, for instance, done so that somebody would have the access for a few days of microscopy through Instruct, but then they've come to my lab, for instance, on an Embl fellowship. And so they're actually working with us for 3 months on the Embl fellowship, but the actual microscopy time is then provided through Instruct. So there are different ways of doing this, and Claudia is always willing to discuss this as well. But personally, I always like to have people in the lab. There was somebody at the back?
Hi, Sarah. So I wanted to ask, the Vitrojet, do you think it might help in the preparation of... Sorry. The Vitrojet, do you think it might help in the preparation of samples that have, for instance, detergents or glycerol or things that usually make it difficult to...
So, there's a lot about the kind of what's happening at the interaction with paper and water and air and grid that we don't know about, and what we're trying to do now is to separate these factors to find out whether or not it's just this blotting which is a problem, or is it more due to the way that molecules distribute within an air-water interface. Our findings so far are that with samples with different types of detergent, we can certainly get nice grids with the Vitrojet. At the moment, I can't say whether we are seeing disruption of particles in the air-water interface. I don't think we've seen any examples of that so far, but on the other hand, we have very, very rarely seen that also with other methods of making grids. What I was hoping that maybe we thought sometimes that if you get a collection of molecules that will sit at the air-water interface, that if you would then bring the blotting paper to them, maybe you would remove a lot. And we thought maybe we'll see differences in the concentration, right, now when we don't have the paper taking things away. Turns out the sorts of concentrations that we use with our blotting and without blotting work about the same. So I don't think we're removing anything, but we're also not concentrating. So it's early days yet. We've only had the machine up and running now for like 2 months. But as we progress, we will certainly spread the information and say. But at the moment, I'm very willing to try things which people have had problems with, because I think this is one way to see if we can get around.
Thank you.
Just one more question,
Hi, thank you very much for the presentation. I was wondering, given your previous explanation, you have solved the issue of orientational bias. Have you seen any differences in conformational bias in using the Vitrojet versus LIGO?
We don't have enough process data yet to be able to say that. And I would need to have a sample which has really got conformational bias in it in order to be able to test that. So if you have one, come and speak to me.
Just a big question. This single cell proteomics, is that really a single cell, or is that just enriched through the labeling?
Marco tells me it's a single cell. I would direct that question to him. It's better than 10,000. Okay, I think we need to move on. Thank you.
And now we will have Christoph Müller from Embl Heidelberg. Embl is also a recent member of Instruct, but we hope to get very much out of the Embl. And Christoph is going to tell us about that. Okay, thank you very much.
Okay, thanks. First of all, I also would like to thank for the invitation. It's great being here at this great venue and to be able to present. So yes, Embl is a new member of Instruct, and so I thought it's perhaps good if I just introduce you a little bit what Embl can offer. And so, as you know, Embl is an international organization with 27 member states by now. And Embl is a distributed organization with 6 sites, and so the main site is here in Heidelberg, but then there are the Embl EPI, Barcelona, Rome, Hamburg, and Brno. And in fact, there are 4 structural biology stations or sites at Embl. There's Embl Heidelberg with a focus on cryo-electron microscopy, as you will hear. There's Embl Hamburg with strong focus on sample preparation and access to Finland, protein crystallography and SACs. And then there's Grenoble, again, high throughput crystallization and access to the macromolecular crystallography beamlines and SACs. And then, very importantly, that's often forgotten, there's also the Embl EBI, which is the databases, the PDB, you obviously all know, but there's also EMDB and Empire, which stores the raw data. And then lately now also there are all these AlphaFold structures with more or less in the future very accurate predictions being available for all these different proteins. And however, today I will really focus on more on the experimental side, and will try to explain what is available there. And obviously, it's not a very long presentation, so please ask also questions. We can also still discuss afterwards. So again, this is the overview on the three sites: Grenoble, Hamburg, and Heidelberg. Grenoble and Hamburg both being located next to a synchrotron site, and I'm sure many of you have already been there. And so just to remind you, at Embl Grenoble, this is the Embl site is next to the ESF synchrotron, which just got upgraded to be a 4th generation synchrotron, but there's also the neutron reactor, and on this what is called the European Photon and Neutron Campus, there's also the IBS, which is also an Instruct Center. And the focus in Grenoble is on one side high throughput crystallization, and then obviously also to allow access to the ESF beamlines with many users, more than 2,000 as you see there, in 2018. And then recently there's also access to cryo-EM in Grenoble through the ESF, together in collaboration with Embl and the IBS. So it is a very rich environment there for integrated structural biology. And now, within the Instruct-Embl, Embl Grenoble offers access to its high throughput crystallization pipeline, so you can send a sample, it will put into high throughput crystallization. It's followed up by optimization of initial crystals, and then it becomes automatically harvested. And I'm sure many of you have already seen that. So this is done using a laser. So the crystals grow on a film, on a thin film, and then a laser comes, and this laser cuts out the crystal where it is, and automatically mounts it. This is all done automatically, and then it gets automatically transferred to the beamline to automated data collection, and in the next step, automated data processing. So the way you send the sample, it goes through the pipeline, and if you're lucky, you can have directly a structure determined there. What is important, or where this is obviously very important for, is for compound and fragment screening. So you can really now screen large amount of, you can screen it against compound libraries, but you can also screen it against fragment libraries, and then quite quickly get insight how a certain target protein binds small molecules, and get the insight there. The next slide just shows you this again, and how this is actually the view on the facility by itself. And this is a very successful operation which has been running for many years with more than 1,000 users, 4.5 million crystallization experiments, and access has always been through INEX first, and then now INEX Discovery. Access is through Instruct, but then also through FRISBI, that's the French infrastructure, the French branch of Instruct. And as I said, it really allows this very rapid compound screening and automated fragment screening, and I strongly encourage you to try it out. These are the heroes. Stephen Cusack, obviously, since many years, and then Rosan Marquez is the person who builds this all up, and as you see, he builds it all up because he's everywhere. He's the scientific contact, he's the technical contact, and he's the administrative contact. You cannot avoid Rosan, but get in touch with him. He's extremely enthusiastic, and will be very happy to talk with you in our community. Okay, now we're switching over to Embl Hamburg. Again, there is integrated structural biology service. Hamburg is very strong on sample preparation and characterization. They have a fantastic facility for biophysical characterization. And once the sample is characterized, then it can either go to the SAX beamline to P12, Dimitri Svergul. Many of you might know his name, and or perhaps already have worked together with him, or collaborated with him, or used his services. And then there are just two macromolecular beamlines, P13 and P14, where again you can collect data to high resolution. I should say special strengths or special emphasis at Embl Hamburg, is dynamics and time-resolved experiments. Beamline is extremely strong, and you can do time-resolved experiments there and analyze those, and they have a lot of expertise answering these kind of questions. And so again, these are the players. There's Matthias Willmans as the head of the Hamburg side, and Thomas Schneider. And it's Thomas Schneider and Dimitri, but Maria Garcia-Alachi is responsible for the sample characterization, biophysical characterization. And again, I can only encourage you to get in contact and visit this site. Okay, now I'll come to Embl Heidelberg. I should say I'm coming from Embl Heidelberg. And so there the focus is really on cryo-electron microscopy, cryotomography, and cryo-clem. And I should say we have, in the time when shortly after the resolution revolution in cryo-electron microscopy, we have been very trying to react fastly to that and to set up an initial platform, and we call this a service platform. And that has worked actually quite well through INEX, and then now INEX Discovery since 2021, when Embl opened its centers, also now through Instruct. But all this has been set up also in preparation of the imaging center, the Embl Imaging Center. This is a whole center which I will introduce to you in a little bit greater length. And so, in a way, this service platform was really more a pilot to start to get to know the requests and the needs of the user community. And this is now extended, and the service has been extended to the new imaging center. And actually, Sarah, you mentioned that in a way we were very limited initially with people. So we were more kind of a service where we say, if you have a sample where you already have rates which look reasonable, we can help you there. But meanwhile, we can now offer much more comprehensive service. I'll show you in a minute. Okay, and so now this is a little bit more about the Embl Imaging Center. So this is a brand new building. The building has been completed in the mid of 2021, and since the fall, we have had pilot projects there and started to get all the workflows ready. And the imaging center is now open, so you can apply it at any minute. You can, once your projects, will explain that there are different access routes, but when it's approved, you can actually get quite rapid access. And the imaging center offers quite a bit of different services and things. So on one side, it's really open access to new imaging technologies, and the ambition is really to go from high resolution imaging like by cryo-EM all the way to organismal imaging, for example, with light sheet microscopy. And a special focus, and I'll repeat that several times in the talk, is really to emphasize also the correlative approaches between these different techniques so that you can connect one with another. There's tailored and expert project support, people there to help you. It also comes with quite a large number of different courses, and in the imaging center, very well known is the Embl cryo course, which has been running, I think, for 20 years already, and is always incredibly oversubscribed. But then there's also, there are commercial partners also in the imaging center, and the idea is really to there's always time gap once especially in light microscopy, somebody sets up a microscope and it works well until there's a commercial product. And the idea is that by having companies being involved there and work together with the scientists, that this innovation is just going on much more rapidly, that innovations much more quickly get translated into products. You can find more information about the Imaging Center on the website. So again, the idea is really to try to go from the analysis from individual molecules to molecular machines, complexes, all the way to cells, tissue, and to organisms. And so, to on one side, understand the structure, high-resolution structure, the architecture of complexes, but also how they function, how the machines move, what is the dynamic of these molecular machines, and ultimately how these machines then function in the context of cells, but then also entire organisms. So it's really a very comprehensive view on this. And obviously one would like to have one microscope which you look into and then you can zoom and you will see everything, but that obviously doesn't happen. You need different instruments and you need the correlative techniques, and that's a particular emphasis of the center. And so, and the center offers on one side sample preparation, so people can come just with their sample, and I'll show you examples of that. And then you can, people there help you in the image acquisition using the different techniques what you use, either high throughput, either cryo data collection or super resolution microscopy, whatever you think, and people also help you in the data analysis. So our mission is really to provide a comprehensive service. And in the following now, I'll go a little bit through what is offered. So first, if you look at the light microscopy, so the light microscopy services, they cover different resolution levels. They work also at different throughput levels. Some of them can be high throughput, and working on image samples with different thickness and exploit different labeling regimes and chemical physical principles. And this, on the left side, you see a little bit the scope. This is actually a nuclear pore where some of the porings have been fluorescently labeled, and so you see here the nuclear pore in two different organizations. These are microtubules again, where you can look at the dynamics of microtubule assembly and decay. And then we go into the organismal imaging, and where, as you can do it by, for example, by light sheet microscopy. And what are the instruments which are available? Well, there are different super resolution microscopes are available. There are more commercial ones which are already commercially available, like the STET microscopes. And then there are a single molecule like location microscopy, super resolution microscopy, which are home-built instruments. And then there's through collaboration with company Embl, which has been founded by Stefan Hell, who got the Nobel Prize for his work on super resolution microscopy. So this is an influx instrument. This is for the highest possible resolution, where you can look at in very high resolution to samples. And then there's the light sheet microscopy, and which is more for organism imaging. On the correlative side, I'll quickly show you there the workflow. So here, a typical workflow is, there are obviously always variation, but a typical workflow is that you have here a cryo-EM grid. You can grow cells on this cryo-EM grid, and then and freeze this grid, and then you can look with cryo-confocal light microscopy, which are really the area of interest, where are the proteins you want to study, which are fluorescently labeled, because even a cell is small, finding the process you're interested in within a cell needs very often additional labeling techniques. And so you can do this with this cryo-confocal microscopy, and you can identify an area of interest. And in the next step, you then use what is called a FIB-SEM, a focused ion beam scanning electron microscope, and this is essentially this uses a gadolinium beam to cut out a very thin sections. These lamella, they have about 200 nanometer thickness, and you need this very thin lamella to be able to analyze them with high resolution cryo-electron microscopy. And so this is done here. So this area which you identified before and where you identified the location is put into this machine and it cuts out a very thin lamella. And then you transfer this lamella into a high-end electron microscopy. Usually it's a type cryos. And then you can do cryotomography where you tilt your sample and you do tomography data reconstruction. And so this is the workflow, and this is something which is working there very well in collaboration with the Embl scientists. And so you can use this workflow. And finally, I'll come to the electron microscopy activities there. So there we provide service in sample purification. So again, you can come with a sample and people will help you using to find the best conditions to make cryo-EM grids. Depending on what you want to do, we have the possibility to do the cryo-fin milling. I just explained that. We do data high resolution for cryo data collection. And there is also the possibility to do volume imaging by cryo microscopy. And these are the instruments which are available. There's a Titan Cryos, soon 2 Titan Cryos in the imaging center available for external uses, for single particle analysis, high-res tomography, and cellular tomography. There's a Classius. We use this Classius to pre-screen samples before we put them in the Titan Cryos, so that we find the very best grids and the very best holes in the grids to do the data selection. And then we have, this is the crossbeam shown here, we have getting our second instrument, both for feed milling, this is this making this singular manner, and then it's the cryo-conformal line management. You might actually notice that it's the other way around, as explained in the previous slide. And these are some of the people who provide the support, like Wim Hagen, some of you might know him, and then these are the people, the team there. And this is just one example to show how that it's actually working. I mean, this is an example during our pilot study. So there was a visitor, Kate O'Connor, she came from the lab of John Atkins from Ireland, and she brought purified 70S ribosome, and this is a very special ribosome. It's a stalled ribosome, so they had a very it's stalled in the translation process, and so there's a very sophisticated biochemical optimization of the sample has been carried out, but they hadn't done cryo-EM. They were in no ways cryo-EM people, so they came with this sample, and we helped her to get this into the cryo-EM grid. It was then scanned using the Classius microscope, and then a high-resolution data, and it turned out that they found very nice-looking grids, and then they transferred that over to the Titan Cryos and collected within 2 days data set on the Titan Cryos, and they obtained reconstruction in 1.9 angstrom resolution, which I couldn't believe this, but I mean, I mean, it's pretty amazing. But I think it also has to do, I mean, just as a disclaimer, I think it also has to do with the fact that it's the stalled ribosome, which is in a very stable conformation. I'm not a ribosome expert, but I think I understand that there are movements there which makes this, in many cases, not so straightforward. But just to say, the pipeline is working, and the pipeline is actually waiting for you in this. And so there are different ways how you can access this. There's access through direct access, so you can pay a contribution to the running. So given the total cost, it's actually a small contribution, I have to say, but there's direct access. Nevertheless, projects get reviewed, but then there's also access through Instruct for a member of Instruct. Members of Instruct. And then there's also access for neurobiology. This is for the Instruct is obviously for the cryo-EM related services and so on, whereas for light microscopy services, it's neuroimaging. And then there's the C findings, which you all know also, which IMEX Discovery and different others now also. IMEX Discovery in the first place. I would, if you want to access, in any case, and you want to send in an application, it's always good to first ask to discuss your proposal, what you want to do with the scientists. They're very happy to help you also to recommend how you apply and so on. And it's always worth doing this. And as I said, once projects are approved, it's actually you can actually have quite rapid access. It's working well. And this is the team hungrily waiting that you apply. I mean, it's a double team, so the person responsible for the cryo-EM service is Simon Matai, he's the team leader for Electron Microscopy, and this is Timo Zimmermann, he is the team leader for the light microscopy imaging. And so, as I said, they are very happy to receive you there. And these is summary of the many important players which provide access to all the different sites as part of Instruct, but then also of the online access.
Thank you very much for a very elucidating presentation on Embl and various services and sites of Embl. And as you see, there is great potential for applications here as well. So if you want to ask any questions, either now or during the day. Sorry.
Yeah, I have a question regarding so first, I'm really glad to have you on Instruct, because it's for me that I'm starting on the cryo-EM and tomography. It's a really nice opportunity to submit the project to you and to be able. So we'd like to know, in your portfolio, do you think, especially because you have a close relation with the synchrotron, you are thinking to also put X-ray imaging, fluorescence imaging on your system, on your.
You mean X-ray imaging?
X-ray imaging.
Well, there's two things. I mean, there's already X-ray imaging activities at the EWSF, and in Hamburg they just recruited this Duke, and she's in the process of also establishing hard X-ray imaging beamline in Hamburg. So both, but this is I think at the moment there it will have to go through the EWSF. It's not part of the experiment.
Yeah, because it will be nice to do the correlative microscopy coupled with X-ray.
Yeah. I mean, this is clearly, this will come. But it's,
Okay, so thanks to the organization for allowing us this slot. And as Maria Eugenia said, we have a very strong collaboration with people in Portugal, and you will see about the talk in a bit. So I will present the Instruct Center in Spain. And how does it go? Yeah,
Point here? Sliga.
I have to go.
Left Sliga.
Yeah.
Sliga no pointer. Já não foi uma colega ligar.
I have PowerPoint.
Experimenta agora. Já está. Já está.
Thank you. So, yeah, this is the National Center of Biotechnology, where the Instruct Center is. Actually, we provide two facilities there. So one is the cryo facility. I don't know if this is important. So we have the cryo facility. We have. So the cryo is where the microscopes are. And then we have the Instruct Image Processing Center, that is where we do the data analysis. And both are in the same building, in the same place. There is a lot of communications. So I will start with the machines. So the cryo facility, it is headed by José María Valcuesta here, but it was started by Rascosa and José María, and these are the people involved. Probably you know some of them, you have it already there. And this is the email if you want to contact them. So we have kind of relatively standard tools to vitrify the samples. We have Vitrobot, a few instruments for data, and then this high pressure freezing is used for relative microscopy and for electron tomography. And then we have several microscopes. The first one is this one, that is 120 kilovolts, relatively standard microscope, low-level microscope, but it is automated, and we can do cryo microscopy there. It is mostly, thank you. So it is mostly dedicated to negative staining and sample screening. And then we have this one, that is the one for the true one for sample screening, cryo sample screening. And this is 200 kilovolts, and we have a faceplate, Falcon 3 direct detector, and also it has automatic loading, and we can do automatic acquisition. Actually, many of the studies they stay here because it gives already 3 ounces resolution, and if you are happy with the structure, then you stop at that point. And if your sample is very good, then it goes to the cryo amp, that is 300 kilovolts, and then it has an energy filter, faceplate, K3, Cat K3 detector, and again it is automated through serial EM, and then you acquire the high resolution data there. And this is not part of the facility as offered in Instruct, but we have also in the same institute, we have cryocorrelative microscopy, we have a FIPSEM equipment, and we are setting up now the microelectron diffraction. And this will come with a SITA camera that will be put in the talus. So, to access this equipment, you can do it either through INEX Discovery now or through Instruct. There are a few differences between the two. The main difference, so they have things in common, like you have to agree with the facility, which is the volume that you are going to analyze, the concentration, the buffer, so all this sample preparation stuff. And then you can do remote access or on-site access also in both places, and then you are reimbursed for the trip if you have to travel. And what is different in INEX Discovery is the shipping to the facility is also covered by the project, and there is a detailed report at the end. And typically the projects start with a screening at the talus article, and then if it is good, the sample is good, then it moves to the cryo arm. And through Instruct, it is almost the same, and even the time, I think it is can be a little bit longer in INEX Discovery. So here it is 3 days, the other one is 3 or 4 days. And then the main difference is this shipping, that now if you are not using the travel spending, then you can use it to ship the sample. And the detailed report, you don't get it. I don't know why they do like this, but it is like that. Then, yeah, so the cryo, so this is the access to the talos. So the access to the cryo, it is again 3 days per sample, and it goes also through Instruct. So you submit the proposal to Instruct. You have to give evidence that you have a good sample, or you have a pre-screening in the talos, and then you go for the cryo. Then, once you have acquired your data, you may go into the image processing center. Also, we accept data acquired anywhere else, so not only in Instruct, but you can also collect by your own in any microscope or whatever. And then you submit your proposal to process this data at the Instruct image processing center. If it is approved, then we start the processing. And in this processing, the people in charge are Jose Maria and myself. And then here, Blanca is the administrative contact, and Marcos and Roberto are the people actually handling the projects. Actually, Marcos has been here, he has collaborated with you quite a lot. Then the resources we have for the processing is, from on one side we have hardware, so this is a very big machine. So it is a collection of machines. And then we also have software. So our infrastructure view is that the software is also infrastructure, and we need somehow to solve how to communicate different programs. We were talking just before lunch that you can go from CryoSpark to Reliant to XB to whatever, and everything inside the same platform, so that it is very convenient, and you can compare the results between the different software. And the pipeline typically is this one. So you submit your proposal, then it is evaluated. So these two things are done in Instruct how the proposal evaluation is done by scientific evaluators outside Instruct. And then once the project is approved, you transfer the data to our machines. This is a remote access that was mentioned before. So for us, it is the routine way of working. And then we have like 2-3 weeks of image processing on that data set, and then at the end we take the decision. So this is the final structure, or this data goes nowhere. This is the maximum that we can achieve with this data. And maybe the decision is to take courses on data. And it has been mentioned a bit, but the proposal is really easy. So if you go into area to make this proposal, it is in total it is something like 2-3 pages, no more than that. So it is really easy to write what you want to have, and then it will be evaluated by the Instruct evaluators. So, kind of summary. So, you have your sampling solution. So, your sampling solution, you can apply to INEX Discovery and Instruct at the same time. So, you could do the vitrification and acquisition of the data through INEX Discovery, and then the image processing, you can do it through Instruct. If you have your vitrified grains and you are shipping your vitrified grains, then you can either apply to INEX Discovery or Instruct. So in Instruct we don't have this, there is a combination that we don't have. But so if the vitrified grains can go through either of the two, and then if you have already your movies, your acquired data, you can simply apply to Instruct for the image processing. And yeah, so you will have to show that you have some preliminary results, and these are the typical things that we ask, that you have a pure protein, and that you have a nice concentration on images, and even better if you have 2D averages, especially if you want to go for the high resolution, you need to show high resolution averages. And then the data is transferred through multiple technologies, so you can choose the one that works in your institute. And you can transfer up to several terabytes, and it may take some time depending on what is the bandwidth at your institute, but at the end, in a few days, all the data is transferred for sure, and then we start processing. And during these 2-3 weeks, what we do is all the image processing pipeline. So starting from the movie alignment and then checking that the micrographs have a good CDF, a good properties of the microscope at the moment of acquisition. Then we find the particles, we align them, and at the end we do the 3D reconstruction. And during the whole process, there is a very narrow communication with the users. So we keep updated about how the project is going. And here are some results, some examples. At the end, these are examples that when the whole pipeline, so the purification and acquisition and image processing, they're in manual. Some examples, some other examples. And then that is something that has not been mentioned, but when you submit your application to Instruct and you acquire the data at Instruct, the data management policy states that the data will be made publicly available after some environment. So after 2 years, it will be made public. At present, it is not done because there is more infrastructure to do that, I think. But at some moment, it will be enforced, this policy. We are very active in training, so every year we offer some courses related to cryo-EM. This year it will be electron tomography. So the electron tomography is within Instruct, but we offer 4 courses during the year: simple particles, streaming, image processing in streaming for facilities, and then atomic modeling and every 3 months. This year, electron tomography is in Instruct. Otherwise, we also participate in Portuguese by we are very happy to be with you here. So I think that is also the main take-home message would be you have free access to state-of-the-art image acquisition and image processing. And the fact that the 2 services are within the same institute, they allow a very nice coordination between the 2, so that at the same time that we are acquiring, we start processing. Then you are getting feedback about how the acquisition is going and whether the sample could be replaced by something else, or if it is very good, and then you will be very happy at the end. But both, so they can be coordinated, but both can also be accessed independently. So you can do whatever you want, whatever better fits your project. So that is all. These are the 2 main mail addresses for both services. And I'm open for questions if you have. For questions. Otherwise, we cannot have.
So these talks, we don't have for discussion now.
Sorry. Carlos, one of the big problems that we are experiencing is the data storage for the dozens of terabytes that are generated. Does your facility has a long-term storage facility capability, or does the users have to take care of their own data?
So as stated in the policy, it is the user responsibility for the long term. So we have something like midterm, like 3 months. For 3 months we can store the data. We have a couple of petabytes at the facility, but this microscope generates 2 terabytes per day, so very easily you fill it. So yeah, the long term is on the user side.
More questions? Christoph, you said there is no resource for storing the data. Isn't that Empire where you can store all this, the raw data, or
The is in the data. So yeah, so we have, for instance, a pilot project with Empire that all projects going through Instruct, they can deposit immediately the data at Empire. And the data is not released, so they have also some embargo period at Empire. And after these 2 years, if they produce the reconstruction, then it is public, and otherwise it is deleted.